University of Paris, and 1794. And he was a postdoctor and researcher in the group of Pierre J. Beckwith at Harvard Medical School, from 94 to 1950. And in 96, he got a position in the Pasteur Institute in Paris and joined in the group of Pierre Guilhot to develop a genetic process to explore the biological and pathogenic of tuberculological bacilli. And he moved for the Institute of Pharmacological and Structural Biology of Toulouse in 2000 to start the molecular bacterial pathogenicity team through the ITP Avenue Young Researcher Program. Since 2001, Dr. Guilhot has become a permanent employer of the Centre National to Research Scientific, and he assumed the position to co-chair of the Department Molecular Mechanisms of the Mycobacterial Infection and member of Molecular Mechanisms of Mycobacterial Infection, and member of IPBS, yes, that's right now. IPBS Executive Committee from 2009 to 2015. He stepped down from his chair's position in 2015 and now concentrated on the pathogenic, on his researcher work on the evolution and mechanisms of pathogenesis of tuberculosis bacilli. His research program continued to be supported by fundings from FRM, National Research Agency, AU, and Bettencourt, MSDA Avenir, and Bill and Melinda Gates Foundations. Thank you, Dr. Christopher, to be with us and make yourself comfortable to start your presentation.
So thank you very much for this very exhaustive introduction. It reminds me that I'm not so young now, and I have been working on tuberculosis for so many years, but how? That's life. So thank you very much. It's my pleasure, and I would like to thank also Dr. Olmo for the invitation to participate to this course. It's really a pleasure for me to discuss with you our current view on the evolution and emergence of tuberculosis bacilli, and also to discuss with you our recent results on that topic. So thanks again for the introduction and for the invitation also. And I'm going to share my screen. I hope that everybody is hearing me correctly. If not, I may at some point put a earphone and a microphone if it's not loud enough. Otherwise, I will continue like this. If there is,
it's totally okay.
Okay, so if you have any question, feel free to ask the questions maybe on the discussion on after the presentation or during the presentation if you want. It's not a problem. So okay. I'm going to share my screen now. Okay, this one. Okay, is it okay for the screen? Okay, fine. So the topic of this presentation is to try to identify and characterize the adaptation that have been associated with the emergence of tuberculosis bacilli from an environmental ancestor. So in fact, The current view of the emergence and evolution of tuberculosis bacilli is that this view is supported by many and numerous phylogenetic studies, is that in fact Mycobacterium tuberculosis ancestor emerged once from an environmental bacteria approximately 10,000 years ago. And in fact, it switched from the environment to a mammal host, probably a human, through acquisition of capacity to persist in this new host. And this is, I mean, this is not specific to Mycobacteria and Mycobacterium tuberculosis. This emergence from environment is something that is very common among pathogens. For example, we have learned that from the COVID, but I mean, it has been known for many years for various pathogenic bacteria. But so the same story occurred in Mycobacterium tuberculosis history 10,000 years ago, where the ancestor started to colonize the human host. And this transition was associated with an increased capacity to persist and to survive in this host. This evolution continued during the history of tuberculosis bacilli, and subsequently the tuberculosis bacilli ancestor acquired the capacity to transmit from one host to another. And this is this acquisition that led to the very efficient epidemic bacteria that we know today. During this evolution, one specific lineage also changed host specificity to colonize other mammals. Mainly cows, but also many other mammals such as opossum, badgers, seals. And during this transition from a human to other mammals, they lost the capacity to transmit in humans. So that's why at the moment there is no real transmission from human to human due to, for example, bovis bacilli. And very recently, in fact, two French scientists also derived from this one animal adapted bacteria, the current BCG that we are using at the moment to vaccinate again Mycobacterium tuberculosis. So as I mentioned, in fact, this emergence from the environmental ancestor, the current paradigm is that this event occurred only once. And after that, the bacteria, the tuberculosis bacilli diverged and evolved, but they evolved clonally without acquisition of new genes from other bacteria, for example, and this generated what we know now as the Mycobacterium tuberculosis complex, excuse me, that gathers 8 major phylogenetic lineages, most of them found only in humans, such as the Mycobacterium tuberculosis sensu stricto bacteria, but also the Mycobacterium africanum. And one group of bacteria, as I mentioned, is mostly found in other mammals, such as bovine, capre, or seals, for example. And those bacteria are still able to cause disease in humans, but they are no longer able to transmit in humans directly from human to human. So all those bacteria are very closely related, and their genome is around 4.4 megabases. And if you compare two genomes from 2 bacteria from this Mycobacterium tuberculosis complex, the difference is less than 2,500 SNPs. So they are very, very similar and very, very closely related. If we go one step back in the tree for the phylogenetic tree for the mycobacteria, the close relatives of the Mycobacterium tuberculosis are whole opportunistic pathogens. So Mycobacterium tuberculosis are obligate human pathogens. The animal adapted strains are obligate animal pathogens, but the close relatives are whole opportunistic human pathogens. That's true for Mycobacterium caniti, the closest relative, which shares with Mycobacterium tuberculosis approximately 98% nucleotide identity. So it's below the limit for 2 different species, the limit being around 95. And So clearly, Mycobacterium mecaniti is from the same species than Mycobacterium tuberculosis. If we go back, there are other bacteria that are very similar to Mycobacterium tuberculosis with average nucleotide identity between 80 to 85%. So they are not per se the same species, but they are very closely related. And all those bacteria are opportunistic pathogens. They form what we now call the Mycobacterium tuberculosis associated phenotype. So in this group of opportunistic pathogens, Mycobacterium mecaniti is very interesting because of its very close relationship with Mycobacterium tuberculosis. And in fact, what is interesting is that if you look at the phylogenetic tree of the Mycobacterium tuberculosis complex there, or the Mycobacterium mecaniti there, as you can see is that the branches are longer for the Mycobacterium mecaniti than for the Mycobacterium tuberculosis complex. It means that the difference between every strains from the Mycobacterium tuberculosis complex is much lower than between every strains from the Mycobacterium mecaniti group. And another interesting observation is that most of the tuberculosis cases in humans are due to this Mycobacterium tuberculosis complex, whereas there are only a few cases of tuberculosis in humans due to Mycobacterium mecaniti, and all those cases are found in people that lived in the Horn of Africa. And if you look at the genomes of these Mycobacterium mecaniti, their genome is a little bit larger than the one from Mycobacterium tuberculosis. It's 4.43 to 4.52 megabases for the Mycobacterium mecaniti in contrast to the 4.4 for Mycobacterium tuberculosis. Their genetic distance is a little bit larger. It's between 16,000 to 61,000 SNP in comparison with any strains of the Mycobacterium tuberculosis complex. And also between strains from the Mycobacterium mecaniti, the distance is also larger than between strains of the Mycobacterium tuberculosis complex. So this is for the phylogeny, for the genomes of those bacteria and for the epidemiology of those bacteria. But if you look at the features and the phenotype of these bacteria, they're also a little bit different. Whereas the Mycobacterium tuberculosis displayed this typical rough colony morphology that you can see on this picture. Those are colony on a Petri plate of Mycobacterium tuberculosis. So they exhibit this very classical rough colony morphotype. This is completely different for the Mycobacterium mecaniti. Here it's one strain of Mycobacterium mecaniti, Mycobacterium mecaniti STBK. And as you can see, you have this kind of smooth colony morphology. So the colony morphology is very different from Mycobacterium mecaniti and Mycobacterium tuberculosis. As I mentioned, their genome is a little bit larger, and the level of pseudogenization is also less advanced in Mycobacterium mecaniti than in Mycobacterium tuberculosis. As I mentioned, there is no described transmission from human to human due to Mycobacterium mecaniti, so they are probably acquired by the infected people from an environmental reservoir. So although this feature led many groups to believe that, in fact, those Mycobacterium mecaniti are somehow not fully adapted to colonize the human host, and they are somehow similar to the ancestor of Mycobacterium tuberculosis. So, of course, the ancestor is no longer around in the environment, no longer in people, but what looks the closer to what we imagine this ancestor is those Mycobacterium mecaniti. So, interestingly, also, in fact, those Mycobacterium mecaniti, as I mentioned, are less able to transmit in humans, and they are also less pathogenic in various animal models. For example, in mice, which is a very classical animal model when we work with Mycobacterium tuberculosis. So when we infect mice with Mycobacterium tuberculosis, here bacilli mice, when we infect with, I don't know, around 100 bacteria, what you observed, if you look at the bacterial load in the lungs, you observed a rapid increase during the first 4 weeks of the infection. This is the acute phase of the infection. So you have this rapid increase. And then you have a plateau that is called the chronic phase of the infection. During this plateau, the bacterial load remained very stable for Mycobacterium tuberculosis. And during this plateau, the bacteria tend to, the animal tend to accumulate lesion in the lung, as you can see on this picture, and at some point the lung will lose its function and the animal will die. If you do the same kind of experiment with the various Mycobacterium caneti strains, what you observed is again a very rapid increase during these first 4 weeks of the infection. There is this acute phase of the infection, but during the chronic phase of the infection, the bacterial load in the lung tend to decrease for Mycobacterium caneti infected animals. And in the worst cases, for example, in animal infected with this specific Mycobacterium caneti STBK strain, the bacteria is even eliminated from the lung. And the lesion is also much lower in the case of animal, in the lungs of animal infected with Mycobacterium caneti. And in fact, their lifetime is much longer than for animal infected with Mycobacterium tuberculosis. So clearly, it seems that Mycobacterium caneti, also very similar to Mycobacterium tuberculosis, is not fully adapted for the long-term colonization of the human host and probably other mammals. So it seems that Mycobacterium caneti, also not exactly the environmental ancestor of Mycobacterium tuberculosis, probably look like the environmental ancestor of Mycobacterium tuberculosis. So the questions we have been interested for a while in my group, but also many other groups have worked on this problem, is what are the adaptations associated with the transition from an ancestor that looked like Mycobacterium caneti? What are the adaptations that are associated with the increased persistence and increased capacity to cause disease and to spread in humans? So this is what we call the emergence and evolution project. And as I mentioned, the question is mostly what are the bacterial adaptations associated with this emergence for the TB bacilli from environmental Mycobacterium caneti-like ancestor. So to address those questions, many groups have been interested to understand that, because it will help to understand the process and the risk also to have or the emergence of pathogenic mycobacteria from environmental mycobacterial species. So one way to address that question is to look at the genomes. And this is something that has been done by several groups worldwide. And they have tried to compare the genomes of bacteria that are not Mycobacterium tuberculosis, but are close relative of Mycobacterium tuberculosis, such as the Mycobacterium tuberculosis associated phenotype strains. And they have compared the genome of those strains with the one of Mycobacterium tuberculosis to try to identify the specific adaptation that makes Mycobacterium tuberculosis such an efficient pathogen. So the group of Roland Broche, for example, at the Pasteur Institute has done an extensive and very nice work to try comparing the genomes of those bacteria with the one of Mycobacterium tuberculosis. And what he has found is that most genes known to be important for the interaction with the host, such as, for example, the toxin-antitoxin genes, but also genes involved in the biosynthesis of specific lipids, or the genes involved in the secretion of various virulence factors, all those genes are all present in these opportunistic pathogens. They are not at all specific to Mycobacterium tuberculosis. So they concluded from this study that, in fact, the ancestor of the Mycobacterium tuberculosis acquired the major virulence function before the branching from the Mycobacterium caneti. In fact, this acquisition occurred at this time point during the evolution of the ancestor of tuberculosis bacilli, and it was way before the colonization of the human host. So it was a kind of pre-adaptation for colonization of a host. Another series of studies try to compare Mycobacterium caneti with the Mycobacterium tuberculosis, because, as I mentioned, Mycobacterium caneti is an environmental bacteria that is very closely related to Mycobacterium tuberculosis. Is the closest opportunistic pathogen than to Mycobacterium tuberculosis. So again, the group of Roland Broche, but also the group of Inaki Comas in Spain, they have compared the various Mycobacterium caneti strains with the many Mycobacterium tuberculosis strains, trying to identify gene or mutations specific to Mycobacterium tuberculosis. And again, in fact, they have not found many genes that are specific to Mycobacterium caneti. In fact, they found only one gene that might be important, the PGRF33, which has been shown in several studies to be important for the virulence of Mycobacterium tuberculosis. So this gene is specific to Mycobacterium tuberculosis complex, and it seems to be important for the interaction with the host. They have found also two phages that are specific to Mycobacterium tuberculosis, but these two phages so far have no demonstrated function for the interaction with the host. There are also several functions that are specific to Mycobacterium caneti, such as, for example, these genes, COBEF, that is important for vitamin B12 biosynthesis. And in fact, Mycobacterium caneti is able to synthesize vitamin B12, whereas Mycobacterium caneti tuberculosis is not able to synthesize vitamin B12. It has to acquire vitamin B12 from the host to fulfill several functions. Another gene that is specific for Mycobacterium caneti is the gene PKS8. It's not functional, it's a pseudogene in Mycobacterium tuberculosis, but it seems to be functional in Mycobacterium caneti, but so far we don't know. In Mycobacteria, and especially in Mycobacterium tuberculosis, PKS family of genes are encoding protein involved in for the formation of different lipids, and some of them very important for the pathogenicity. In the case of PKS8, we still don't know the real function of that gene and the encoding enzyme. So clearly there are very few genes that are either specific to Mycobacterium tuberculosis or Mycobacterium caneti. The group of Inaki Comas in Spain, they have looked a bit more carefully to try to identify mutation or genes that are different in terms of sequence in Mycobacterium tuberculosis than in Mycobacterium caneti. And actually they found 53 genes that are more divergent in Mycobacterium tuberculosis than in Mycobacterium caneti, suggesting that mutation in those genes might have been important to acquire the capacity to colonize the human host. Also, it has not been demonstrated so far. Another genes that were identified by the group of Inaki Comas as may be important for this transition was the gene 4R. And in fact, they found that in Mycobacterium tuberculosis, 4R is under positive selection, whereas in Mycobacterium caneti, it's not under such positive selection. So positive selection means that it tends to accumulate mutation. And so far it's unclear why 4R in Mycobacterium tuberculosis tends to accumulate mutation. But clearly that's something that is clear from their comparative genomic studies. So the consequence of this mutation acquisition is not clear at the moment. So the main conclusions from these comparative genomic studies are that the transition from environmental bacteria to obligate pathogens was associated more with adaptation of endogenous pathway rather than gene acquisition or loss. So in my group, we decided to use another strategy, which is more a kind of experimental strategy to address the questions of the adaptation that are important for the epidemic capacity of Mycobacterium tuberculosis. So the first way we tackled this problem was to try to analyze why Mycobacterium tuberculosis displayed this different colony morphology in comparison to Mycobacterium caneti. So this is again the picture of the colony on plate of Mycobacterium caneti versus the colony on plate for Mycobacterium tuberculosis, with the typical rough colony morphotype there and the smooth colony morphotype there. And it's also associated with a different behavior in liquid culture, with lots of aggregation in the case of culture with Mycobacterium tuberculosis, whereas we observed a more dispersed culture in the case of Mycobacterium caneti. So these features suggest that the surface of the Mycobacterium tuberculosis and the surface of Mycobacterium caneti are different. And when you are talking about surface, you're of course talking about the cell envelope of the bacteria. And the cell envelope of the Mycobacterium tuberculosis is very unusual and very complex. So here I have just put a few pictures of the electronic micrograph pictures of the cell envelope of Mycobacterium tuberculosis and the scheme of the organization about the various layers of this cell envelope. And again, a scheme of the lipids specific to this outer layer. So as you can see, in fact, in Mycobacteria and also in Mycobacterium tuberculosis, the cell envelope is made of two lipid bilayer. Above these two lipid bilayer, there is a capsule, and this capsule is responsible for the colony morphology and the property of the surface. If you go deeper in the structure of this cell envelope, in fact, you have this plasma membrane there, which is very similar in its composition to the plasma membrane in other bacteria. What is unusual in this is this outer layer and the outer membrane of the Mycobacteria. This outer membrane in Mycobacteria is mostly made of mycolic acid. So those are very long chain fatty acid that are attached to a polysaccharide, the rabinogalactan. This forms the inner leaflet of this outer membrane. The outer leaflet of the outer membrane is made of, again, mycolic acid that are attached on 3 halos, and they're also made of unusual lipids that are more or less specific to the mycobacterial species. For example, in Mycobacterium tuberculosis, within this outer leaflet, but also in the capsule, you have a whole range of lipids that are specific to Mycobacterium tuberculosis. Those polyacyl trialos, the sulfolipid, or the diacyl trialos are all specific to Mycobacterium tuberculosis. There are also several other lipids that are not specific to Mycobacterium tuberculosis, such as the phenylglycolipid or the fuserol dimycoserosate, that are also found in Mycobacterium tuberculosis, but they are also found in other mycobacterial pathogens. And finally, there are this lipo-oligosaccharide that is found in many mycobacteria, but usually not in Mycobacterium tuberculosis. So if the surface of Mycobacterium caneti is different from the surface of Mycobacterium tuberculosis, we reason that maybe it's because the composition of the cell envelope is different. So we analyzed the composition of the cell envelope of various Mycobacterium caneti strains and of the lipidic composition of the cell envelope of many different Mycobacterium tuberculosis strains from the various phylogenetic lineages. And so we analyzed the various lipids, and the obvious difference was the capacity to produce lipo-oligosaccharide. As I mentioned, usually Mycobacterium tuberculosis is not able to synthesize lipo-oligosaccharide. However, all the Mycobacterium caneti that we analyzed are able to synthesize this lipid. And this is shown here on a thin layer chromatography plate where the glycolipid were revealed with antron. And as you can see, here you have the spot corresponding to the lipo-oligosaccharide, so they have different migration, probably due to different structure, but all the Mycobacterium caneti are able to synthesize these lipids. In contrast, here you have one strain of Mycobacterium tuberculosis, but it's true for all the strains of the Mycobacterium tuberculosis that we analyzed. They are unable to synthesize this lipo-oligosaccharide, and you have the structure of this lipo-oligosaccharide there. So it seems that a main difference between the Mycobacterium caneti and the Mycobacterium tuberculosis are the capacity to synthesize this lipid. So to go further and to try to analyze the impact of this difference in the colony morphology and the ability to synthesize lipo-oligosaccharide, my colleague and friend Roland Broche and I, we started a project, and this work was mostly done by Eva Boric in his group and Vladimir Malaga in my group. And in fact, we isolated from Mycobacterium caneti 2 strains of Mycobacterium caneti, the strain K and I. We isolated spontaneous mutants that display a rough colony morphotype. So that was quite easy because those mutants occurred at relatively high frequency. So you just have to plate the bacteria and you look at the colony morphology and you can see that some do not display the parental smooth colony morphotype, but they display more this kind of rough colony morphotype. So we isolated several mutants that either from the genetic background from the I Mycobacterium caneti or the K Mycobacterium caneti, and we performed genome sequencing on those mutants, and we analyzed the genome in comparison to the parental genomes of Mycobacterium caneti K or I. And in the case of the rough mutants derived from the high Mycobacterium caneti, we identified several mutations in 2 genes, PKS51 and PKS52, which are located in the same locus. And in the case of the rough colony morphotype mutant derived from STBK, again, we obtained a mutation in the same locus, but it was a recombination between the 2 copies of the PKS5. And this recombination led to deletion of this enterogenic PAP gene. So we next analyzed the impact of this mutation on the production of lipo-oligosaccharides. So we analyzed the lipid production in, for example, here you have the colony, the impact on the aggregation in liquid mediums. Here you have the rough colony morphotype of the STBK mutant, and here you have the parental STBK. But we also generated a complemented strain in which we reintroduced this locus in the mutant, and this restored the smooth colony morphotype and the ability to grow dispersed in liquid medium. So we performed lipid extraction and we analyzed the lipids in the mutant and in the wild type and in the complemented strains, and the results was very similar for the two series of strains. The wild type was able to synthesize lipo-oligosaccharide there and there. In the case of the IR mutants, it was less able to synthesize lipo-oligosaccharide, and when we reintroduced the wild type copy of this locus, we restored the production of lipo-oligosaccharide. This effect was even more pronounced in the case of the R mutants derived from STBK, where we were no longer able to detect any lipo-oligosaccharide in those mutants, and when we complemented with the functional copy of this and the wild-type copy of this locus, we restored the capacity to synthesize lipo-oligosaccharide. We also analyzed by GCMS the fatty acid that are specific of these lipo-oligosaccharide, and here you have the results for the rough mutants. As you can see, the 2 fatty acids that are specific to the lipo-oligosaccharide are no longer found in the rough mutants derived from STBK. So clearly, those mutants that displayed this rough colony morphotype are not, or either less able or no longer at all able to synthesize lipo-oligosaccharide. So the next question was, oh yeah, and interestingly, I forgot to mention that, but it's very important. If we looked at the genomes of Mycobacterium tuberculosis, in fact, the organization of the genome is very similar to what is found in the R mutants of STBK. In this region, in fact, there is a single copy of PKS5, whereas there are usually 2 copies of PKS5 in the Mycobacterium caneti, and there is no longer this PAP gene in Mycobacterium tuberculosis. So it seems that what's happened in Mycobacterium caneti STBK-R mutants is similar to what's happened in the ancestor of the Mycobacterium tuberculosis strain. So, of course, the next question we were interested in is what is the impact of this loss on the interaction with the host? So to address that question, we used the series of strains derived from STBK with the parental smooth variant, the rough mutant and the complemented strain. And we infected various infection models. We used cell lines such as human THP1, we used guinea pigs, and we used the skid mice. So as in all those models, what we found is that the rough mutant was more able to multiply and to cause disease and to kill these mice. In the human macrophages, as you can see, the rough mutant is able to multiply, whereas the smooth variant is less able or not at all able to multiply, as well as the complemented strain. The same was true for what you see in guinea pigs, where the bacterial load in the lung of the infected guinea pigs was much higher after 42 days of infection for the strain, the animals infected with the rough mutant in comparison to the smooth parental strains or the complemented strains. And the same was true also for the ability to cause disease in the immunocompromised skin mice. Here you have the time to kill curve, where you have here the curve for the animal infected with the rough mutants in comparison to the time to kill curve for the animal infected with the smooth parental strains or the complemented strains. So as you can see, there is a delay associated with a lower capacity to cause the disease. So clearly in those three infection model, the rough mutant is more pathogenic than the parental smooth variant. When we switch to a more resistant animal model, here the bacilli mice, the situation was quite different. And in fact, we were unable to detect very strong phenotype for the rough mutant in comparison to the smooth parental strain. So clearly the results from this study was that the transition from Mycobacterium mecaniti like ancestor to Mycobacterium tuberculosis was associated with a recombination at the PKS5 locus and the deletion of this bad gene with lost capacity to synthesize lipooligosaccharide. And this change in colony morphotype due to this genetic event was associated with an increased capacity to colonize and to multiply in hosts that are susceptible to infection with Mycobacterium tuberculosis. However, in hosts that are more resistant to infection with Mycobacterium tuberculosis, the impact of this mutation was mild or not so obvious. And at least this mutation was not responsible for the increased capacity of the Mycobacterium tuberculosis to persist during the chronic phase of the infection. So to go a little bit further, we decided to use another strategy and another experimental strategy. And in fact, we reasoned that if Mycobacterium mecaniti is less able to persist in the lung of the mice, if when mutants with enhanced capacity to persist emerge in this population, it should outcompete the population, and it should be fixed in the population. So we decided to implement an experimental evolution strategy by passaging Mycobacterium mecaniti in mice to try to select mutants with enhanced capacity to persist. The idea was to mimic what's happened during this transition from the environmental ancestor of Mycobacterium tuberculosis to the bacteria that's able to colonize the human host. So we did that in mice, and this work was done in my group by very talented people. I did a postdoc in the lab with the help of Vladimir Malaga. And what they did was to start with two Mycobacterium mecaniti strains, STBK and STBD. STBK is the more distant from Mycobacterium tuberculosis, and STBD is the closest relative of Mycobacterium tuberculosis. So the idea was to try to sample what's happening in these two very extreme situations. And what they did was to infect mice with 10 to the 4 bacteria, to let the infection go for 56 days, and then they recovered the bacteria from the lung of these infected mice. They grew a culture, and they did another infection. So for that, they passage, in fact, 4 different evolution lineages, and none of these evolution lineages were mixed during this experimental evolution experiment. And for Mycobacterium caniti STBK, they performed 15 cycles, and in the case of STBD, they performed only 6 cycles. So in fact, in the case of STBK, the population was passage in mice for 3 years, and in the case of STBD for a bit more than 1 year. And for every passage in mice, what they did also was to measure what we called the persistence index. This persistence index is the log ratio of the bacterial load at day 56 over the bacterial load in the lung at day 28. So in the case of STBK shown here, this persistence index is -2. So whereas in the case of Mycobacterium tuberculosis, as there is no decrease during this chronic phase of the infection, the persistence index is 0. So they monitored this persistence index over the cycle, and as you can see on these 2 panels, for STBK and STBD experimental evolution, this persistence index increased to reach a plateau very close to 0, so very close to the situation for Mycobacterium tuberculosis. This was true for STBK and true also for STBD. So this result meant that our experimental evolution selected population with increased capacity to persist in the lung of the bacillus. So this was true at the level of the population, but we wanted also to test whether it was true at the level of individual clones. So we picked individual clones and we tested them individually. And here you have the results of one such experiment where we tested 4 clones obtained after 9 passage of the STBK. And we compared the capacity of these clones to multiply and to persist in bacilli mice in comparison to the parental STBK or 1 strains of Mycobacterium tuberculosis. And as you can see, during this acute phase of the infection, all the strains are able to multiply and approximately at the same level. But during the chronic phase of the infection, whereas Mycobacterium tuberculosis persists as expected, Mycobacterium caniti display a decrease in bacterial load over the time. The situation for the 4 mutants was much more similar to the situation in Mycobacterium tuberculosis, with 2 clones very similar to Mycobacterium tuberculosis, and here 2 clones intermediate between Mycobacterium tuberculosis and Mycobacterium caniti. So clearly, our experimental evolution selected population and clones with enhanced capacity to persist in the lung of mice. So of course, the questions was, what are the mutations associated with this higher capacity to persist? So we sequenced the genomes of several hundreds of clones derived from this experimental evolution, and we analyzed the SNP that were fixed in this population over time. And the situation was very simple in the case of STBK. In fact, mutation in only 2 loci were fixed independently in the 4 evolution lineages. The first mutation to be fixed occurred in the RV1339 locus of Mycobacterium tuberculosis, and in fact, it was fixed very rapidly in this population. This was a point mutation in the autologs of genes RV1339 from Mycobacterium tuberculosis. Later, during this experimental evolution, other mutations were fixed independently in the different evolution lineages. And interestingly, the mutation occurred in the same locus, the PE18, PPA26 locus, but the mutation that were fixed in the various evolution lineages were different. They were either, in the case of lineage C, a deletion of the entire region, in the case of lineage A, a friendship, a deletion in PE18, and in the case of lineage B, an insertion in PPE26. And again, these mutations were fixed in the entire population between cycle 9 and cycle 15. In the case of STBK, the situation was more complex because in the initial population, we had a mixture of different variants. We had what we called the major variant that was present at 60% in the population, but we had also other variant in the population, a variant that was called V30 because it was present at 30% in the population, and another variant that we called V3% because it was present at 3% in the population. And curiously, in fact, those variants do not display the same capacity to persist in mice and the same fitness ability in the competition, because very rapidly after 3 cycles in these mice, the major variant in the initial population was almost completely replaced by the V30 variant. Interestingly, in fact, other mutations were fixed in those population in various genes. And interestingly, in lineage C, also, although this variant had almost disappeared after 3 cycles, probably one mutation occurred in this variant that make the strands very efficient to persist, and it was again re-enriched later during the experimental evolution. And this mutation was probably in this gene. So to go further and to try to understand the impact of this mutation and the interaction with the host, we decided to focus on the situation in the STBK because it was much more simpler than in the case of STBT. So we decided to work with 2 mutants, KC9A1 and KC9C1. KC9A1 that contains only 1 mutation in RV1339. And Cas9C1 that contains 2 mutations, one in RV1339 and the deletion of the P18, PP26 genes. The first question that we asked was, are those mutations important for persistence only in BALB/c mice? Indeed, we did our experimental evolution in BALB/c mice, so we selected mutants and population more adapted to survive in BALB/c mice. But are those populations specifically adapted to BALB/c mice, or they are more adapted to survive in general in mice and probably in mammals? So to address that question, we switched to another mouse model, the C3HBFEJ mouse model that is now very popular in the tuberculosis community, because this mouse model mimics very closely the lesion found in humans after infection with Mycobacterium tuberculosis. So now it's more and more used to investigate those pathogen interaction in the tuberculosis. So we infected those mice with either the STBK or the Mycobacterium tuberculosis, or those 2 mutants, and we compared the ability of those mutants to persist for long term in those mice. And here you have the results after infection with 10 to the 3 bacteria, and we looked at the bacterial load in the lung after 76 days of infection. And as you can see, whereas we have a low level of bacteria for STBK and a much higher number of bacteria for Mycobacterium tuberculosis in the lung of these mice, the situation for the 2 mutants were in between, but much higher than for STBK. Here we have approximately 2 logs increase in bacterial load in the lung of the mice infected with those 2 mutants. So clearly the mutation and the population that we selected in BALB/c mice had acquired mutation that provide advantage in various hosts, including the C3H host. So the next question was, is there any difference in the histopathology lesion induced by those 3 strains? So we analyzed the histopathology, and here you have a picture of the lungs of mice infected either with Mycobacterium tuberculosis or Mycobacterium caniti STBK, or the 2 mutants KC9A1 and C1. And what you can see in purple are the cells the immune cells that enter the lungs and tends to accumulate if there is an inflammation. So as you can see in the case of the mice infected with Mycobacterium tuberculosis, you can see this very nice lesion with lots of inflammation, formation of caseous lesion that is typical of tuberculosis. In the case of the mice infected with Mycobacterium STBK, the number of lesions was very similar than for Mycobacterium tuberculosis, but the surface of inflammation was much lower. And in the case of the 2 mutants, and surprisingly, the level of inflammation was very similar to what it was observed for STBK. And when we quantified this, this is shown on this panel, as you can see, the level of inflammation for the 2 mutants are very similar than the level of inflammation for the STBK and much lower than the level of inflammation for Mycobacterium tuberculosis infected mice. We also looked at the cells that accumulated in those lesions, and what we found is we looked at B cells, T cells, macrophages, and neutrophils, and what we saw is that lesion containing Mycobacterium tuberculosis tends to accumulate less B cells than lesion of mice infected with Mycobacterium caneti, and the same was true for the 2 mutants. In contrast, the lesion of mice infected with Mycobacterium tuberculosis tends to accumulate and attract more neutrophils than lesion of mice infected with STBK or the 2 mutants. In all those cases, the 2 mutants tend to behave more like the parental STBK than Mycobacterium tuberculosis. Although in the case of the more evolved mutants, it tends to get closer to Mycobacterium tuberculosis. So the main conclusion from this part of the study was that the increased capacity of the 2 mutants was not due to a change in the immune response, because the immune response seems to be very similar to the one induced by STBK. So if the reason for the higher persistence was not due to a different immune response induced, we reasoned that maybe it could be due to a higher capacity to resist the impact of this immune response. And if you look at the different stresses that Mycobacterium tuberculosis has to face in vivo, there are numerous. This is due because Mycobacterium tuberculosis is able to invade different kinds of cells, different tissue, and even in the lung, they are able to induce many kinds of lesions. Here you have just a series of pictures from human infected by Mycobacterium tuberculosis, and you have different kinds of lesions going from nascent granulomas to caseous necrotic granulomas, fibrocaseous granulomas there. And each of these lesions is associated with different kinds of stresses. For example, in the early granulomas, Mycobacterium tuberculosis is believed to multiply inside macrophages, so it has to face acidic pH, oxidative nitrosative stress, and various nutrient deprivation or metal intoxication. In the case of the late-stage granulomas, such as the caseous granulomas, most of the time they are hypoxic, they are either acidic or neutral. There are different kinds of stresses and toxic peptides also found in those granulomas. So in fact, to survive in those granulomas, Mycobacterium tuberculosis has to have many kinds of strategies to deal with those multiple stresses. So we tested several stresses to see whether the mutants that we selected were more able to survive those stresses. So we tested many different stresses in vitro, and for most of them, we were unable to see any difference between the 2 mutants that we worked with and the parental STBK. This was true for the acidic pH, phosphorus-depletion, H2O2 stresses, for example. However, in some cases, we were able to identify stress that affected more the parental strain than the 2 mutants. This was true, for example, for zinc intoxication. When we grew those bacteria in medium containing high amount of zinc, we observed that the parental wild-type strains was more affected in its growth than the 2 mutants. This effect was even more pronounced when we replaced the zinc by copper, And as you can see, with the 500 micromolar of copper in the medium, the parental STBK is no longer able to multiply, whereas the two mutants are still able to grow. However, for those two stresses, Mycobacterium tuberculosis seems to behave more like STBK than the two mutants. For example, with 500 micromolar of copper, the Mycobacterium tuberculosis is also no longer able to grow. However, when we looked at, for example, susceptibility to nitric oxide, the situation was different. So we incubated back the two mutants or the parental strains or the Mycobacterium tuberculosis with 5 millimolar of nitric oxide, and we measure the surviving bacteria by plating this culture and counting the number of colonies. And after 48 hours of treatment, as you can see, the number of surviving bacteria for STBK is much lower than for Mycobacterium tuberculosis. And in fact, the two mutants seems to be also more resistant to nitric oxide than the parental strain. So clearly, it seems that the mutation in those two mutants are associated with an enhanced capacity to resist stress associated with the immune response. And in the case of this nitric oxide phenotype, it seems that the reason for this increased capacity is associated with the RV1339 mutation, because this mutation is common for the two mutants. And in fact, we confirmed that by both mutation transfer and complementation analysis. So the next question was whether this enhanced capacity to survive and to survive nitric oxide treatment was specific to the STBK or something more general. So we first looked at various Mycobacterium caniti strains, and we repeated the experiment to treat Mycobacterium caniti or Mycobacterium tuberculosis with nitric oxide at 5 millimolar, and we monitored the surviving bacteria after 48 hours of treatment. And again, as you can see, for all the Mycobacterium caniti that we tested, the level of survival was much lower than for Mycobacterium tuberculosis. We next looked at what's happened in the STBD experimental evolution. So we tested 4 clones derived from STBD after the experimental evolution, and 2 of them acquired also an higher capacity to resist nitric oxide in comparison to the parental strain. And finally, we looked at what's happened in Mycobacterium tuberculosis. So we tested different strains of Mycobacterium tuberculosis for their ability to survive nitric oxide stress, and we found that all the strains of Mycobacterium tuberculosis that we tested behave more or less like the Mycobacterium tuberculosis H37RV that we used in all these experiments. So clearly, it seemed that a major difference between Mycobacterium caniti and Mycobacterium tuberculosis is this ability to survive nitric oxide. And our experimental evolution somehow selected this higher capacity to resist nitric oxide. So the next question was, what are the molecular mechanisms behind this higher capacity to resist nitric oxide? So, and to survive in mice. So we analyzed more carefully the mutants that were selected during this experimental evolution. And in fact, we found 2 pathways that seems to be very important for the persistence in mammals. The first pathway is the ability to acquire nutrient. And in our experimental evolution, we found many mutations in both experimental evolution of STBK and STBD that affected the ability to uptake or to use different carbon sources. First, we found mutation in the PE18, PPE26 that was recently demonstrated by the group of Cliff Barry to form porins important for uptake of various nutrients such as glucose, glycerol, or salts such as phosphate and magnesium. We also found a mutation in the MCE1 transporter system. We found mutation in that genes that is important for the MCE1 transporter system that is involved in uptake of oleic and palmitic acid. And finally, we obtained many mutations in GLPK that is involved in that is encoding a glycerol kinase involved in activation of glycerol for its use in the either glycolytic to produce energy or in the biosynthesis of various lipids. And interestingly, when we went back to the work by the group of Inaki Komas, where he analyzed he compared the genomes of the Mycobacterium canity and the Mycobacterium tuberculosis, he found that the MCE1 operon also acquired many SNP that are specific to Mycobacterium tuberculosis in comparison to Mycobacterium canity. So it meant that what was observed during the emergence of tuberculosis bacilli was also mimicked in our experimental evolution. The second pathway that was affected and mutated in our experimental evolution is a pathway leading to the production or degradation of cyclic AMP. So cyclic AMP is a very important secondary messenger in every bacteria, but it seems to be especially important in Mycobacterium tuberculosis. Here, what we obtain is one mutation in a gene that encodes an alylate cyclase important for the biosynthesis of cyclic AMP. And we also obtain mutation in the gene that encodes the phosphodiesterase involved in degradation of the cyclic AMP. And as I mentioned, in fact, cyclic AMP is important in every bacteria. It's a very common secondary messenger. But in Mycobacterium tuberculosis, it seems to be especially important for the interaction with the host. Indeed, whereas, for example, in E. coli, which is the classical laboratory strain of E. coli, there are only two, to my knowledge, there are only two adenylate cyclases. In the case of Mycobacterium tuberculosis, there are more than 16 different adenylate cyclases that are involved in the production of cyclic AMP. And those adenylate cyclases are recognizing signals from the host or the bacteria, and upon recognition of these signals, they produce cyclic AMP. And this cyclic AMP is then important for upregulation of various genes important for copper resistance, but also virulence or use of various carbon sources or production of siderophores for the uptake of iron. So clearly, it seems that cyclic AMP metabolism in Mycobacterium tuberculosis is a key pathway for regulation associated with survival within the host. So we are here in our project of the emergence of tuberculosis bacilli. So what are basically the lessons for all this work on Mycobacterium tuberculosis evolution and emergence of tuberculosis bacilli from what probably a Mycobacterium canity-like ancestor? The main lesson that is that first, a change in bacterial surface associated with the lost capacity to synthesize lipooligosaccharide occurred in the ancestor of Mycobacterium tuberculosis, and probably this mutation was a first step for the increased capacity to multiply intracellularly and to survive in the host. Other key pathoadaptation differentiating Mycobacterium tuberculosis from their environmental ancestor are this higher capacity to survive stress encountered within the host, and especially nitric oxide. And also the ability to uptake various and to use different carbon sources, such as the one found in mammals, such as lipids, for example. Another lesson from this Mycobacterium tuberculosis evolution project is that when we did that in lab, in fact, in our experimental evolution, or even in vitro for the lipooligosaccharide, it was quite easy to select for mutant with higher capacity to both multiply and to survive in the host. So this is somehow surprising if you consider that the emergence of tuberculosis bacilli occurred only once from the environmental ancestor. If it's so easy to acquire this capacity to survive in the host, in mammals, and to persist, for example, in humans, but also in other mammals, if it's so easy, why this event occurred only once? And in fact, if you consider this evolution, increased persistence capacity is the first step. If you want to colonize a host for long term, you have to acquire the capacity to transmit and to transmit efficiently. And this is probably something else that is missing in Mycobacterium mecanity. And in fact, Mycobacterium mecanity, even if it's able to produce some tuberculosis cases in human, it has never been described to be able to transmit in humans. So probably something is missing in Mycobacterium mecanity, something else than the ability to persist in the human host. And again, this increased capacity to transmit could be related with the change in the hydrophobicity of the Mycobacterium mecanity during this transition. Because several groups showed that in fact the capacity of the bacteria to be aerosolized, and it is very important for the transmission of tuberculosis because the transmission is via aerosol in tuberculosis, and the capacity to be aerosolized, nebulized in the air is associated with the hydrophobicity of the bacteria. The more hydrophobic the bacteria is, the more easy it is to put it in aerosol. So maybe the change in hydrophobicity, such as the one associated with the loss of lipooligosaccharide, is associated with an enhanced capacity to be aerosolized. Another maybe important adaptation is the ability to synthesize sulfoglycolipid. Indeed, very recently, the group of Tim Schillow in the US, they demonstrated that the capacity to synthesize this very specific sulfoglycolipid that is produced by Mycobacterium tuberculosis, the capacity to synthesize this sulfoglycolipid is associated with induction of cough. And cough is very important for the production of droplets that are associated with transmission. And when we looked at the production of sulfoglycolipid in Mycobacterium mecanity, we found that most Mycobacterium mecanity are unable or less able to synthesize sulfoglycolipid in comparison to Mycobacterium tuberculosis. So here you have the sulfoglycolipids. Here you have 4 strains of Mycobacterium mecanity. All of them are less able in vitro, at least, to synthesize sulfoglycolipid. There is one exception, which is STBJ. So we don't know at the moment whether this different capacity to synthesize sulfoglycolipid is associated with an increased capacity to induce cough, but that's something we are very interested in. And with that, I'm going to stop there. And I would like also to thank the people in my group. And here you have my group in Toulouse. So it's a very nice group of people that I have the chance to lead. All the work that I have showed you there, and at least the part of the work that was done in my group was produced by these 3 people, Heidi Nalin, Catherine Astari de Keker, and Vladimir Malaga. All those people have left the group at the moment. They are now working somewhere else. And I have also the chance to collaborate with very numerous people, including Roland Broche at the Pasteur Institute in Paris, Philippe Suppli at the Pasteur Institute in Lille, and several other groups worldwide. And again, I have also the chance to be supported by many funders that help us to perform these studies. And with that, I'm going to stop, and I will be pleased to answer any questions that you may have. If there are any parts that have been unclear, feel free to ask, and I'll be pleased to answer. Thank you very much.
Thank you, Dr. Guilhot, for your brilliant presentation today. And the students, if you have any questions, you put on the chat, please. We have a first question here on the chat. Yes, and please open your video or open your audio if you want, students, and make you feel free to ask if you want it. Just open your audio and, you know,
So do you want me Do you want me
Him,
to
if
take
you
directly the
wanna.
questions from the chat? Okay, so there is one first question from Dr. Olmo about RV1339 and its ability to modulate autophagy. We have absolutely no idea at the moment. We have not looked at autophagy in infected cells by mutants of RV1339. So that's something we may look at, but I have no results about that, so I don't know. Maybe another questions from Flavio Alves. Which copper salt did we use for the? So it was not for the Ross experiment. It was we used copper salt for copper intoxication. I don't remember. I have to check. I don't remember. I will send you if you. Yeah, I don't remember. I'm sorry. So I have to check. I don't know. I don't remember. And if you are interested, you may send me an email, and I will answer you. Oh, hello, Flavio. I remember you.
Hi, Professor.
Nice to
Thank
see
you
you
very much for this presentation. Very nice.
Yeah, I'm sorry. I don't remember. It was. I don't. I am going
So
to.
I misunderstand the data. I think that you are using copper to produce Haas by
No.
Haas
No.
reaction.
No. We used for the Ross. We use hydrogen peroxide. And no, for the copper, we just monitored the toxic effect of copper. So.
And the mutants are also more resistant to hydrogen peroxide.
It was we had lots of problems to really see a difference for the mutants with regards to hydrogen peroxide. It seems that there is a weak effect of the RV1339 mutation on hydrogen peroxide resistance, but it was very. The data were very variable, and it was very difficult to clearly demonstrate that. Whereas for example, nitric oxide, it was much more clear. So, but it's possible, but I'm not sure.
And the entry in the macrophage by mannose receptor are the same. They enter the same. They are uptaken at the same rate by macrophage.
Yes, I would say yes. I have to remember the results, but the results that I had in mind, we did not see any difference in uptake nor in replication in macrophages, at least for a short period of time. But we used human macrophages that are derived from monocytes, and they are very permissive for Mycobacterium tuberculosis replication. So maybe the situation would have been more dramatic in, for example, mouse bone marrow derived macrophages that we may activate to, for example, to increase the stress on the bacteria. But with regards to uptake, from the results that we have, I have not seen any difference between the mutant and the wild type.
Thank you.
You're welcome.
And Dr. Leila has a question. She raised her hand.
Christophe, thank you very much for the very nice talk.
My
it's
pleasure.
Always very stimulating to listen to you. I have two questions. One regarding the mutations in the deletions in the PPE for the. Oh, I wrote it down here somewhere.
Yeah, it's the PPE 26
and
PPE
18.
deletion. Yeah.
They are just one base deletions. I mean,
in fact, we got 3 different mutations in this region. We got large deletion due to a combination of 2 PPE genes, PPE25 and PPE27, that are on both sides of the PE18, PPE26 genes. So in fact, this recombination led to a deletion of the PE18, PPE26, but we also got 2-point mutation that are either an insertion in the PE18 or a deletion of one base in the PPE26 that generate frame shift mutations. So in fact, we got, it seems to me that those two genes that probably worked together, PE18 and PPE26, it seems that the two are important for the persistence of Mycobacterium canity.
But those out of frame mutations, are those genes, do they have coupled translation or no?
Yes, well, from the data published, we have not looked at ourselves, but from the data published, it seems that they form an operon, at least for the PE18, PPE26. But for example, we reconstructed the deletion in another background of Mycobacterium canity, and we were able to also observe in this other background of Mycobacterium canity this increase in the persistence capacity with the real deletion. So it seems that by deleting those two genes, it produced the same phenotype as the small frame shift mutation.
Yeah, because maybe the frame shift has the same deletion effect.
I agree.
It's small. I like those.
It's probably the case, yes.
Yes, and the second question, we are working more with not with TB, but with MLEPRI genes. But the question also applies to TB. When you show the structure of the surface and the capsule, proteins that are anchored to the membrane, and they have variable repeats in the extracellular portion, like PG, PGRS, that kind of protein. So when you, and also, PG, from the days at Brigitte Schickel's lab, you remember that well. So if you shorten or if you increase the number of repeats without taking the N and C terminal, without taking the whole protein out of frame, what do you envisage could be the impact on the location of the extracellular part? It would be embedded in the capsule, do you imagine? And if you decrease the number of repeats by VNTRs, what could that? Did you ever think of those situations? I was wondering if
good questions. I have never thought about that. What would be the impact of the number of repeat?
Yeah, because if you shorten, if a protein is linked to the membrane, has a transmembrane anchor, and it's just has it's the case of would be the case in ERP, which was actually secreted, but anyway.
But behind your question, do you think that, for example, accessibility to the surface would be important in that case?
Yeah, I was just wondering, because we often forget to look at the surface of the bacteria in that way, with the membrane, the capsule, and all that. We have a tendency to think that if something is outside, it's exposed, but in mycobacteria, that's often not the case. It's exposed, but limited in the capsule, so it maybe has a structural more. Anyway, I was just,
No. I mean, it's an interesting question. I mean, of course, I have not the answer. In fact, that's why the problem is that we have not strong rationale to, when we started, for example, the experimental evolution, one main reason to start this experimental evolution was that based on comparison of genomes, there was no obvious, in fact, a candidate, with the exception of the PGRS33, but there was no other very good candidate to study to explain the difference in the persistence capacity of the Mycobacterium canity and tuberculosis. And with regards to the number of repeat, I don't think that there are so many differences between Mycobacterium canity and tuberculosis with this regard. So for us, it was difficult to really raise from this comparison a clear picture, or to identify clear candidates to work on. So that's why we decided to have this kind of unbiased approach to see whether by selecting mutants, we may identify function and genes that are really important. So, but we have not found, well, from our sequencing, doesn't seem that there are any difference in the number of repeats, for example, in the number of, in several of these PE or PGRS. The only mutation that we got was in this PE18, PPE26 region with regards to those family of genes and proteins. So it seems that there is something specific for those genes. But yeah, I mean, we still have to work to really decipher the exact molecular mechanism of and the exact function of these two genes.
Yeah, very interesting. Thank you very much, Christophe.
Thank you. It's nice to see you.
It's nice to see you too.
Dr. Cristina Pessoloni have a question, but first I want to ask her something, and the same line for Dr. Leila Mendonça, because I'm working some time in Mycobacterium abscessus. Is not the same line for tuberculosis and Carnegie, but I'm working with abscessus, Mycobacterium abscessus, and I'm working with a timeline for patients. We have the first, we get the first bacteria, the first infection, the time for first, and we collected different time points of the patient, and they started to, the colony started to change the morphology. And this changing start to make the move for the roof, and we saw the genetically changed for porine and some GPLs on the membrane. And I'm thinking when they used to see this changing for the roof, yes. And it's not, and Leila asked about the changing from the colony is not something in the membrane or the capsule. It's not, could be something in GPL or another things in the membrane. It's not something more down in the membrane or just something inside it, because they can move it and can not something inside can enter in the mycobacteria. And I mean, the patient can, I mean, of course, in the last time point, the patient died. So, I mean, the treatment is not effective anymore. And the treatment was more, make more pressure for the patient with, or the infection or something like this. You see something in that bacteria?
Well, I'm not sure that I got completely the questions, but what you mentioned is that during infection of a patient with Mycobacterium abscessus, there is this switch that is well described from the smooth colony morphotype to a rough colony morphotype. And in the case of abscessus, as you mentioned, it's in most cases related to the capacity to synthesize glycopeptidolipids, so which are not exactly the same than the lipooligosaccharide, but you may imagine that the impact with regards to hydrophobicity and the ability to produce bacterial interaction is the same. Yeah, I think that the parallel is very good. I think that what we observed in Mycobacterium abscessus is very similar to maybe what's happened in Mycobacterium mecaniti. So far, it's because we know that in abscessus, the rough colony morphotype are more virulent than the smooth colony morphotype. And yeah, it's something, maybe it's something very similar to what's happened in Mycobacterium mecaniti during the evolution and emergence of tuberculosis. It seems also that there are differences in the ability to produce and secrete some lipoprotein, no? In the case of the rough variant, no, of the Mycobacterium abscessus. Is that right or not, if I'm wrong? So it seems that the immune response is also completely different with the rough variants of Mycobacterium abscessus, sorry, than for a Mycobacterium, the smooth mutant. Is that right? If I remember correctly. So maybe there are several things associated with this change in composition of the cell surface and the cell envelope. First, of course, the hydrophobicity and maybe the capacity to generate, for example, biofilm or stuff like this. It could be also related to the capacity to induce a different type of immune response. And it seems to be clear for Mycobacterium abscessus, whereas in the case of the Mycobacterium caniti and our experiment, it does not seem to be so clear that there was a different immune response induced by the rough morphotype. Yeah, it could, I mean, it's a major change for the bacteria to have this change in the surface. And in the case of abscessus, the ability to secrete also lots of different lipoproteins. So yeah, that, yeah. Yeah, I don't know if I answer correctly the questions, but yes, I think that I am very also interested with Mycobacterium abscessus because it seems that it's also a bacteria that is in the process to emerge and to change from environmental bacteria in some cases to bacteria able to transmit directly in humans. So yeah, we are very interested also to look at what's happening in these bacteria.
Yeah, thank you for.
You're welcome.
And Dr. Cristina Pesoni have a question. She will ask you now.
So very nice, your talk. Very nice meeting
Thank
you.
you. It's nice to see you.
Yeah, nice to see you too. Yes, very nice. And just curious about your project, and very exciting, really very exciting, but very complex. Difficult to find out which is actually the direction of these mutations. Like I was looking at your results in the terms of your mutants, and they look like they persist better than the wild type. And in terms of Mycobacterium tuberculosis, they produce less pathology. So they look like they are very nice, you know, more adapted pathogens to these mutants. They transmit well in mice. Did you check the transmission?
Well, this is a main problem in the field, is that there is no good transmission model for Mycobacterium tuberculosis, and mice are not able to transmit Mycobacterium tuberculosis. So it's something that, yeah, of course, we'd be very interested to test, but there is no good model to test that. We looked at we have not looked at the release in for example, bronchoalveolar lavage. So I don't know whether the number of bacteria released in the lung are the same for mice infected with Mycobacterium tuberculosis and mice infected with Mycobacterium caniti and the evolved mutants. That's something that I would be very interested to look at. Yeah, with regards to the lesion, it was quite disappointing because we expected to get something closer to Mycobacterium tuberculosis, but I can see two explanations. First, maybe it's not enough. Maybe they are more able to persist, but they are not yet at the level of Mycobacterium tuberculosis. And when we looked at, to support this, when we look at for example, the cells in those lesions, there is a trend. If you look at the mutants, in fact, the mutant is the more able to persist is also the one that looks more like Mycobacterium tuberculosis in terms of different cell population in the lesion. So it seems that the more the bacteria is able to persist, the more it looks like Mycobacterium tuberculosis. So maybe we are still missing something a little. we should increase a little bit again the ability to persist to get to the level of Mycobacterium tuberculosis. So this is clearly one possibility. The other possibility is it could be, for example, related to the PGRS 33 that is not synthesized by Mycobacterium mecanity, and it seems that is an important antigen. And maybe we should try to transfer it to Mycobacterium mecanity and see what's happened, and or maybe delete it to Mycobacterium tuberculosis and see whether now the lesions are closer than the one we get with even the more evolved Mycobacterium mecanity. Yeah, but I agree with you. I mean, it's the lesions are much less pronounced for the Mycobacterium mecanity, even the one that are able to persist, than for Mycobacterium tuberculosis. Yeah.
Yeah. So in this, your last phrase, says that these mutants are better, more evolved in terms of when you compare with Mycobacterium tuberculosis, since they can persist and they don't induce so much pathology, you know.
Yeah, but transmission is associated with pathology. So to transmit, the TB has to produce those kinds of cavities and necrosis lesion to be released very efficiently in the lung. And I think that even if they are able to, persistence is one aspect that's probably is deficient in Mycobacterium mecanity, but there is also this other aspect, which is the ability to really transmit. That has to be, TB has to produce at some point some lesion to transmit efficiently.
Okay, thank you.
So they are more adapted to persist, but less to transmit.
Yeah. Okay, thank you very much. Very nice.
And questions? More questions? No? Any students? Don't be shy. No?
But for the students, if they have, if they want to ask questions, they may send me an email also, or either directly, or you may transfer them my email, and I will be pleased to answer those questions later if they have some. Or if they have some very good ideas also, I'll be pleased to hear those ideas.
Thank you very much. It was a very nice presentation and a nice discussion that you have here this morning. And I would like to thank you for every person that stay with us. And I would like to inform that to do the World Cup, today will not have our program at afternoon, because as I said, Brazilians are very worried about World Cup. So we have a change in our program. And I don't know if you can see, but the presentation of Dr. Bruno Andrade will be on November 29. And I would like to invite you to stay with us. And we'll have Dr. Tânia Silva at 9 a.m. And after Dr. Vinicius Fava, that will talk about HIV and susceptibility to tuberculosis, and also Bruno Andrade that will talk about tuberculosis, diabetes, comorbidity. It will be a very nice opportunity to discuss with these three younger researchers that are waiting for you to stay here to discuss with them. And it will be a great pleasure for us to have all of you there with us. Thank you very much. Thank you, Dr. Christophe, again, to stay with us.
Thank you. It was a pleasure. And hello also to all the people.
Okay, it's a very nice opportunity to extend, to know what our colleagues are doing, and I have a lot of ideas, and I will send you an email,
Okay.
Dr. Okay. Thank you.
I'm looking forward to see.
Okay.
Thank you very much, and good luck for the Brazilian team.
Okay. And for France also. Bye-bye.
Okay.
Thank
Bye-bye.
you. I will close this section now. Bye-bye.
Bye-bye.